So I got an email yesterday about the test today. Yeah, what does that say? If you're ever absent, what should you be doing? Listening to the recordings. Okay, so, moving on. For Module 1, we still have one more set of objectives. It's kind of small, and I have a PowerPoint for it. I just haven't posted it yet because I'm still doing my final tweaks to it. But I did add the reading. It's not a lot of reading left. So it's Chapter 3, Sections 1 and 4. But one of the things I do want you guys to do as we're preparing for next week's test on Monday is one of the objectives listed here is to compare prokaryotes versus eukaryotes. So I made a little quick table of the major characteristics that you should be able to know the similarities and differences as it relates to prokaryotes versus eukaryotes. And notice too, as it relates to prokaryotes, we actually have two domains, right? Both bacteria and archaea. And then the cell type eukaryotes is only the domain eukarya. So we're still continuing on our discussion on prokaryotes, which includes both bacteria and archaea right now. So for homework, see if you can write some things in here. I'm not going to collect it or anything, right? But it'll be a good tool to help you study. So then later today, hopefully, I'll post the PowerPoint, the last one, and the last Connect assignment. No, Wiley Plus, sorry. I'm still editing it. And I'm going to try and figure out how to make it show you guys the answers. I looked back at the other ones, and apparently I can't change them. So I might just post like an answer key or something for you guys to make sure you... But you can do it an unlimited number of times, so I mean, eventually you could figure out what the correct answers are, right? Make you work for it. I think I'll set it at like 3 attempts for this other one, right? Again, I want to make you work for it. So let's see. So that brings us to today's lecture. We're still on the topic of prokaryotes. I hurt my back this weekend. I'm doing better than yesterday, but I'm in a lot of pain right now. So if I seem a little disconnected, and that's why the work didn't get done, because I actually went to bed at 9 o'clock last night. It's really kind of early. I think I couldn't figure out what went wrong, but I think when my dog went to go chase after another dog, kind of pulled my arm and pulled my shoulder and my back, some muscles back there. So they're a little unhappy. Yeah. So. Huh? No, she's not very big, but I wasn't expecting her to run. I wasn't prepared, so she went, and I went. I didn't fall or anything, thank goodness, but she did pull me pretty good. Okay, so let me try to remember where we're at. I think we were just finishing up about how you could destroy peptidoglycan, right? And we talked about the lysostaphin, right? It only really affects Staphylococci species. I was trying to think, did anybody remember for your reading what organisms produces that? I think it's another bacteria that produces that against staph. Produces that chemical that can destroy peptidoglycan. I should know this. This is a new one for me too, though. So needless to say, if I don't know it, it's definitely not on the test. Because it's new. But it's okay. It's kind of cool, though. It's cool stuff. But lysozyme is definitely not new. We've been knowing about this for a really long time, and our bodies actually even produce it, right, in our tears, our saliva, our mucus. and it breaks apart the sugars, right, in peptidoglycan. So I think we just were about to start this, right? Yeah. So there are some antibiotics, and so these are chemicals made by other microorganisms against other microorganisms. So typically you take antibiotics when you have what type of an infection? A bacterial infection. And for instance, penicillin is produced by a mold known as penicillium. And so why would that mold produce something that could potentially kill bacteria? What are they fighting over? Food and nutrients. We talked about this before, right? Way back at the beginning of this course, weeks ago. Right? So they produce these chemicals and they're fighting against each other, and we just have basically stolen their technology. So there's a whole class of drugs that interfere with peptidoglycan synthesis. This whole class of drugs, the reason why we have the chemical structures drawn out is they all contain this ring structure that's referred to as the beta-lactam ring. And there's lots of different penicillins out there, right? We've chemically modified penicillin. So things like amoxicillin, right, are also penicillin, this main thing with stuff added to it. And so what it does is it prevents peptidoglycan from forming the cross links between the sugar layers. So let's look back at the structure for a second. So I want a more detailed. So here, right, the building blocks are made inside the cell and then they're transported out. And you see these bridges right here, like these linkages. It stops those from being able to form. And the reason for that is it actually binds with something inside the cell. So when the cell is making this new cell wall, like if it's been damaged or if it's getting ready to divide, it can't make it structurally sound. It's like stacking a whole bunch of bricks and not putting any mortar between them, right? So you could just walk up and knock that wall over, couldn't you? Without any mortar. So the same thing here, like they'll still stack everything up, but it's not getting linked together. So what happens, right, is that they have these weak points and the cell cannot resist the osmotic pressure anymore and it bursts. Because that's, remember, one of the things that the cell wall does, other than help it hold all those shapes that we've talked about, like bacillus and cocci and spirillum and spirochetes, it helps it with osmotic pressure. So penicillin really only works on actively growing and repairing cells, right? Unlike lysozyme, which will literally just eat away at the cell wall, right? Because it's an enzyme that just goes in and cleaves the bonds between the sugars. So wait a minute. This is a big hot topic for us right now. What about antibiotic resistance? How is it that some bacteria, when we take these beta-lactam drugs, they're resistant to it? It has no effect on them. They continue growing. They just laugh at us. Well, a lot of them, unfortunately, have acquired the information on how to make a very special enzyme known as beta-lactamase. And you can guess where it gets its name. It's an enzyme that breaks the beta-lactam ring. I was too lazy to take my laptop out this morning. So I'm going to add something to one of these slides in a moment. So some bacteria have this enzyme, and they'll destroy any beta-lactam drugs, like penicillin. So remember I said, if it ends in -cillin, it was penicillin, but it's been changed. So augmentin, right, is got this added group to it. Augmentin is combined with a drug. This is not an antibiotic, but a drug. Clavulanic acid. This drug right here. These two are combined in a medication. Anyone know the name of it? You may have taken it. If you work at a veterinarian and you get bit by an animal like what happened to me, this is typically what they're going to give you. No. They're going to give you antibiotics for any infection because, trust me, the wise tale that dogs and cats and human mouths are clean, well, that human mouths are dirty and dogs and cats are not, is bullshit. Okay? Every single mouth is disgusting. Every single mouth of every single animal is loaded and teeming with bacteria. Period. So if you get bit by a human or some other animal, trust me, you want antibiotics. So, augmentin is the name, right, of this combination of amoxicillin and that drug. So I bet you've heard of, how many of you guys have actually heard of augmentin? Heard of it? Taken it? Well, because again, it depends on whether the bacteria are sensitive to it or whether you even had a bacterial infection to begin with. Oh, I don't know how they came up with the word. What augmentin is this antibiotic, amoxicillin, and this drug. So that way, if they have beta-lactamase, this drug deactivates that enzyme and allows the amoxicillin, to still work. Make sense? Yeah, so they're taking care of the problem of if they've got beta-lactamase, this drug eliminates that. Amoxicillin then is able to work because the bacteria is not destroying it. So they put these two things together, right, in the drug known as augmentin. So when they give you the drug augmentin, they're giving you an antibiotic, amoxicillin, and a drug, however you say the name of that acid. It's too much for me. Does that make sense to you guys? Okay. Yeah. Yeah. It stops them from destroying augmentin, right, by having that other drug there that inhibits the enzyme beta-lactamase. Now, we'll talk about it later, but there's lots of different ways that bacteria can be resistant to antibiotics. But this is one of the most commonly known resistances out there, right, is having this enzyme that destroys this class of drugs. It's very, very common, unfortunately. So back around to gram staining, right? So remember the steps you need to know, right? Crystal violet first, iodine, decolorize usually with ethanol, right? And then counterstain with safranin. This was developed by Hans Christian Gram. That's why it's called the Gram stain. It's named after him. And he found when he followed these steps, he got two distinct colors. And we now know that this is based on the differences in the cell walls, or you could even say the cell envelope structure. Remember, the cell envelope is the cell membrane, and then anything outside of that. So if they've got a cell wall, if they've got an outer membrane, all of that is considered the cell's envelope. So for gram-positive cells, we know, especially if you're talking about bacteria, they have a very thick peptidoglycan layer. In addition, because they have so many layers of that sugar stacked on top of each other, linked together by those amino acids, that they tend to have teichoic acid or lipoteichoic acid, which is highly negatively charged, associated through the layers and even attaching to the membrane. That's the lipoteichoic acid. So this is almost like rebar, right? It helps support those layers, just like we put rebar in concrete to help support we have found out recently, they do have a little bit of a space between the cell wall and the plasma membrane. That's referred to as the periplasm. In contrast, gram-negatives have a much thinner layer of peptidoglycan as compared to gram-positives. They have a huge periplasm space that the peptidoglycan is actually said to be in. And they have an additional membrane. So they have an outer membrane in addition to the cell membrane. This outer membrane is important because the very outer leaflet is not made of phospholipids like most membranes. It's made out of lipopolysaccharides, commonly abbreviated LPS. And of course, its name pretty much tells you what it's made out of, right? Lipo referring to fat, poly meaning many, saccharide sugars. So when we zoom in on this component is really important as it relates to gram-negative cells. And you might be like, well, why would my doctor want to know if I have a gram-negative infection versus a gram-positive? Well, structurally, we already know they're different, right? So they're going to behave differently. They're going to be affected differently by different antibiotics. And for gram-negatives, this component, this lipopolysaccharide, is toxic to us. As these cells are dying and breaking apart, this can elicit a very strong inflammatory response from our body. No, inflammation. Fever is different. Fever is an increase in core body temperature. Inflammation is where white blood cells are rushing to that area to defend against invaders or damage that's happened to the body. The problem with that is, how do white blood cells get to where they need to go? What superhighway are they traveling? The bloodstream. So you get that when everyone's experienced inflammation, right? What happens? It gets red, right? That's all the blood rushing there, right? Carrying those white blood cells there. It gets hot because your blood is hot, right? And it gets swollen because the leaky fluid that leaks out of your blood vessels as the white blood cells are traveling out. The problem is you don't necessarily want that happening, like, say, all over your body. What if you get a gram-negative infection in your blood? That's really bad, right? So that's why it gets really scary with gram-negative infections, because even as we're trying to kill the bacteria, they could elicit such a strong inflammatory response that it's actually damaging to the body. So that lipid A component is toxic to us.
Is it possible for the body to attack itself?
Yeah, that's called autoimmune disease.
No, I'm the gram-negative, like disease or something?
Infection.
Then you die.
If you cause too much damage, yes, you could die. Depends. It's one of those broad terms that's used for multiple things. Yeah. Septicemia is when you have an infection in your blood. And that's really dangerous and extremely life-threatening. Most people have a 50-50 chance of surviving, even with treatment. So E. coli is a gram-negative bacteria, right? So it has this lipopolysaccharide. And in fact, there are different strains of E. coli, some of them that are extremely pathogenic to us. One of the ways we can help identify them is, in addition to the lipid A component in the core polysaccharide, they have additional sugars referred to as the O side chain, O meaning opposite of the lipid A component. So you may have heard of the strain of E. coli known as E. coli 0157, right? The O refers to this O side chain. 157 refers to that particular designation for that particular strain of E. coli. So that particular strain of E. coli, what we have designated 157 is the arrangement and the groupings of sugars on their outer membrane. So we have antibodies that can specifically detect that arrangement, what we call 157. So they can test specifically that strain that has those groupings of sugars on its surface. So it's beneficial to us in that we could help, tell one gram-negative from another based on these arrangements of the sugars. The bad news with that strain that we're going to continue to talk about throughout the semester is it produces a toxin similar to another bacteria known as Shigella that causes shigellosis, a really bad intestinal infection. This strain of E. coli also produces that toxin, and that's why it's pathogenic. If you had it, you would not be in this classroom right now. You'd be in the bathroom of the hospital. And we've had several outbreaks of this, right, where food has been contaminated. That's why most people are aware of E. coli 0157. Yeah, I'm not surprised. It happens. Might be. Okay, so gram-positive peptidoglycan layers are really thick, but that layer is more like, imagine a wicker basket, right, that you can put your Easter eggs in, but you certainly wouldn't put water in it because the water would go right through the holes in the basket, right? But it's structurally sound, right? It's a shape. You can put large things in it like eggs. So the same thing is true for the cell wall, right? It's structurally pretty sound, but there are huge gaps in it. And remember, we talked about what happens to these gaps when you gram stain, and in that decolorization step, you do what? You shrink those holes, right? And you trap in the crystal violet iodine complex. So stuff can easily get through that cell wall, right? It's not a barrier that stops stuff from getting in and out. It creates structural support, and especially when the membrane is being pushed up against it because of osmotic pressure. It helps keep it not from bursting. But it's not a permeability barrier. Stuff goes in and out of it very easily. Membranes, on the other hand, we know are permeability barriers, right? They're semi-permeable. They control what gets in and what gets out. So gram-negative, we just said, have how many membranes? 2. They've got their cell membrane and then this outer membrane with all this, sugar stuff sticking off of the ends too. So they have a unique problem in that they have to get stuff across basically 3 layers, although their peptidoglycan layer is so small it's almost not there. So they have to go through the outer membrane, through the periplasm space, and then through another membrane, the plasma membrane. So they have specialized proteins that gram-positives don't have that are embedded within that outer membrane. They're known as porin proteins and the TONB proteins. So porin, that one really helps you, right? Pretty much tells you it creates a pore, right? So that stuff can get through. So these proteins allow stuff to move through that outer membrane. And then, of course, there's other proteins in the plasma membrane that would facilitate the movement from the periplasm space into the cell. This other one, the TONB, is a little bit more complicated. It actually spans both membranes. This is really beneficial, this outer membrane, in being more selective than the plasma membrane. So a lot less stuff gets into gram-negatives that's toxic for them. So some of the antibiotics that we try to use, like straight up penicillin, it can't even get past the outer membrane to affect the cell. So those modified penicillins like amoxicillin, ampicillin, they've been modified in such a way that the cell actually takes them in. No, for some reason, the modification, the cell thinks that it's something it needs, and it takes it in. Takes in the antibiotic, and that's why it's able to work. So, for getting stuff out, too, they have a unique system than any other cells. They have this system that's commonly referred to as the type 3 secretion system. And so within the plasma membrane, right, they have these set of proteins that help facilitate the movement of stuff out. But then they have another set of proteins that create like this tube through the periplasm space. Oh, wait a minute. The whole matrix goes right through the cell, but then they have another tube that can actually insert into other cells. So it's like a little, I mean, when you look at these on electron micrographs, they look like little hypodermic needles. It's really kind of freaky. So these cells, so this is the cell, right? It has this protein matrix that can push stuff right out of the cell and then through a tube that can insert into another cell's membrane and directly secrete stuff into another cell. So as you can imagine, this is very beneficial to pathogens, where they can literally ride up to one of our cells, stick their little syringe in, and shoot stuff into our cells. And there are some pathogenic strains of E. coli that have this secretion system. And they can inject stuff into our cells and cause changes in the cells in our intestinal epithelium. So those microvilli that you have there, they actually form little pedestals, little platforms that the bacteria can attach to and then gain entry into our cells. Evil, scary little things. So they believe that this ability is a modification of how flagella work in prokaryotes, because flagella have this component within the membranes, especially for the gram-negative through both membranes. And when they build their flagella, they make the flagellin protein inside, but then they shoot it up through the tube, and the tube keeps growing. It forms this helical tube of proteins to form the flagella for prokaryotes. So they think that this system just modified, instead of forming a flagella, it stayed a hollow tube where stuff can travel through from one cell to another. Kind of cool. So remember, right, as we said, the alcohol, we believe, shrinks the pores of the peptidoglycan for the gram-positives, helping trap in that crystal violet iodine. Where this step is the differential step, right, something different happens for gram-negatives. Instead, we believe maybe that outer membrane is either removed or quite damaged. That peptidoglycan layer is so thin, it can't trap in the crystal violet iodine, so it gets washed out. And then we counterstain them with safranin so they appear pink under the microscope. Those of you guys in lab will be doing this next week, right? You'll actually do a gram stain. So at one time they thought only the gram-negatives had a periplasm space because their space between those membranes is pretty big. Or you could even say between the cell wall and the cell membrane. And so there's an interesting mini paper in your book where they did some different techniques to be able to study and look at gram-positive cells in a different way, and found that they do have that gap too. It's just not as big. So that gap between the plasma membrane and the cell wall. So next we go to endospores. And again, they only cover it in one little section in your book, unfortunately. So we're going to go into a little bit more detail than your book on this. So for this, we need to be able to identify the structure and what's its function of endospores. That's produced by some bacteria, right? Not all bacteria produce it. And in fact, there are two genera, one we've already talked about, that contain a lot of species within that genera that produce endospores. So what disease did we learn about at the beginning of this course that produces endospores? What's the name of the organism that causes that disease? Bacillus anthracis. What's the genus name? Bacillus. The first name is Bacillus, right? But when we say a species, we say the genus and the species. So we say Bacillus anthracis. So one of the genuses you already know. Bacillus. And then what is sporulization and what is germination? And the crappy thing about all those terms and all the terms we use to describe this process, it all sounds like reproduction, doesn't it? Because probably at one time scientists really thought it was a form of reproduction. But in fact, and this is important, it is not a form of reproduction. It's a dormancy state for that cell. So it's a dormancy. They're just going to chill until conditions become good again. So what's a bad condition for a bacteria? Why would it want to form a spore, basically become metabolically inactive, package up its DNA in such a way that it stabilizes it? I mean, I'm talking about like the best bubble wrap in the world, right? This is the most protected DNA you can imagine, because that's the key, right, in a cell is DNA. If you've got intact DNA, you can do and be anything that that cell needs to be. So that's what it's doing. It's protecting its DNA. It's not doing anything else. It's not metabolically active in any way. Why would you do that? Why would you hibernate? Why do bears hibernate? Lack of nutrients, right? So for them, there are other things that will affect them. Definitely lack of nutrients. We employ this all the time in the microbiology lab. This is why I was in here on Monday, because we're used Bacillus cereus this week. Had I inoculated those tubes on Friday, they would have ran out of food by Tuesday. And so we would have been looking at spores instead of bacteria. and I wanted us to look at the bacteria instead of the spores. So I had to come in on Monday. Yeah, the holiday, I was here. And inoculated tubes. So they wouldn't run out of food. So they would stay bacteria and not turn into spores. So if they run out of food, they're going to form spores, right? They can sense their environment. They're like, oh, it's not looking good. We need to hibernate, right? We need to do sporulization. We need to form a spore. What else would be bad news for an organism? What would be a harsh or poor condition other than lack of food? Temperature changes. What else? You guys said something else? Lack of oxygen or any other gas they may need, right? So gaseous changes. What else? Predators. I don't know if they necessarily could sense that in all honesty, but that would make sense. What else? Water. Everybody needs water. Everybody needs water, right? And the really strange thing about this, though, and the scary thing for us, not for them, is that process of packaging that DNA is a dehydration process. So they actually dehydrate themselves in the process. So once a spore has formed, we can't kill it with plain old boiling. You can't dry it out. It's already dry to begin with. Most chemicals can't penetrate the spore coat that they put around and protect that DNA in. Ultraviolet light can't penetrate it. So how do we destroy these so they can't make us sick? An autoclave, high pressure steam, or certain forms of radiation that can penetrate the spore coat. And there are a few chemicals out there that can actually, but they're pretty powerful, mean stuff. So he's sitting pretty, right? I mean, they have found spores they believe that have been around for hundreds of years. Conditions become good, like you inhale those spores or they get into a wound, because that's the other one. We get, every 10 years you should get your booster for tetanus, right? Does anybody know the name of the bacteria that causes tetanus? It's Clostridium tetani. That's your second genus, is Clostridium. Clostridium difficile. Some of you guys have heard of that one, right? Causes what they call C. diff in hospitals. So when they get into a good environment, you inhale them, they get into a wound, they sense that it's a good environment, they're going to do what's called germination. They're going to turn back into that cell they once were. And they're going to actively grow, right, and reproduce by binary fission. The bad news for us is some of those organisms, like Bacillus anthracis, right, are extremely damaging to us. They attack us, they eat us aggressively. They may produce toxins, like Clostridium tetani produces a very powerful toxin. And in fact, the vaccine you get is against the toxin and not even the bacteria. You can get the bacterial infection even if you've been vaccinated. The vaccine is going to save you from the damaging effects of the toxin it produces. You'd have to take antibiotics to kill the bacteria. The good news is it's gram-positive and standard old penicillin will take care of it. Hasn't it gotten any resistance yet that I'm aware of? Does that make sense to you guys? So as you can imagine, they're kind of terrifying for us, right? Because there are some pretty mean bugs out there that can just basically go dormant when conditions are bad. And it's really hard to kill them or deactivate them. Yeah, long, long time. Because they're not doing anything. They're just sitting there. Waiting to be activated. Okay. So, what's on the outside is probably going to have to stay for next time. So we'll finish, we should definitely be able to finish this up. I hope. The next PowerPoint that I'm going to post is going to be really short for eukaryotes. But again, try and do that table, right? Do that comparison, and I'll also have up, hopefully by sometime tonight, the last Wiley Plus.